Okay, my friends, here we go again. This just came in on Facebook, Live Science. Scientists have determined a giant skull from an ancient human relative named the Dragon Man is actually Denisovan. Well, I have, I'm sure, bigger than what they're talking about, and mine are DNA tested, and they are Homo Sapien, which is modern human mitochondrial DNA. Well, okay, my friends, before we even get started, this is a big discovery. This just came out today. Dragon Man's skull belongs to a mysterious human relative. Now, they've been working on this for, I don't know how long. But this is the skull. Now, I don't know if this is exactly it or is a recreation. I don't know. But it says a skull found near Harbin, China belonged to an extinct line of archaic humans. Look at the size of that brow. Now, we know that there's all kinds of different body styles of humans. There are people with big brows right now, and totally different look to them. Does that mean that they're totally different animals? Now, this was a giant skull as well. All right, it's just coming out now. Dragon Man DNA revelation puts a face to a mysterious whatever. And 20 hours ago, scientists have determined that a giant skull from an ancient human, a Dragon Man skull, may be the first from an enigmatic human cousin. Well, I have stuff also like Notoes, and I have giant human fingerprints, fingertips, and giant human hair follicle. Well, I don't know if it's human, but it's giant, and it's a hair follicle. It has a rectopili muscle, sebaceous gland, root ball, hair shaft. Shown these things a bazillion times, but because they upend everything, this is not a big deal, really. This is just a variation of a human, more or less, although the mitochondrial DNA takes it out of the Homo sapien range. My stuff is Homo sapien. My stuff is modern human mitochondrial DNA, and absolutely stunningly large. Hugely large. A 3-foot long fingertip. Still has the fingerprints and the fingernail. And I went deep inside, took the blood, excellent quality. I'm going to show you all this, or have. So this is not some huge deal. The stuff I'm showing is a huge deal. And the Notoes are also a huge deal. These are feet of creatures that are nothing like us now. And that's the hair follicle right here, and this is what a hair follicle is. I show that so many times because it's easy to show. It's 2 inches wide. It's substantially larger than ours. And this is the feet. We got lots and lots of these. And these are the toes and so forth, and it's just, they're heavily eroded, yes. But this one here is not even eroded at all. It looks like a brand new. It looks like it's a shoe. But it's actually a foot. It's a whole entire foot. And there was no shoe on these. And this is one of the spring-loaded feet. No bones. 2 springs. This one loads up, and then that one loads up with it, and it just drops right back down. That's the heel bone. That's the strap that leads up to the tibia, which is the main bone you stand on. And here it is, right here. That's the heel. That strap runs right up and would attach to the bone, which is what you see right there. There's the heel, there's the strap coming up, it attaches right to the bone. Very easy to see when you look at it. And I've shown that many times. It fascinates me. This is mine, obviously. Well, this is the one that's right here, which is mine. This was from Tish Egerton. She found a whole ton of these things. And that's another one from Tish. And the fibula falls off, just like the fibula is off on this one, too. The fibula is the second bone. It just hugs up against your main bone, so you can sort of twist around and do this sort of stuff with your foot. But they fall right off. And that's a foot. I have it here, and I'll show you a little closer. But those 2 springs load up one on top of the other and then they automatically come back down. It's a very good design, actually. Okay, they're getting closer and closer. This is Dragon Man giant skull, belongs to mysterious human relative. And at long last, scientists have nearly completed the cranium from hominids known as Denisovans. This just came out 18th of June of this year. So let's see what they have to say. Okay, my friends, the walls are coming down. This is from Science magazine. These are big shots. Dragon Man skull belongs to mysterious human relative. Now, and this is a giant skull. And I got into this by somebody sent me this from Live Science, another pretty big shot thing. First ever Denisovian skull identified thanks to DNA analysis. Scientists have determined a giant skull, and it's good size, huge brow ridges. They call it the Dragon Man. It's actually Denisovan. Well, let's talk about the different species of humans. And what about my Notoes? Where do they fit into this? These are creatures that were like humans, only they had no toes. You know, they had a shoe-like foot, and even had almost like a sole built into it, but it was driven by springs. We have these, I have a lot of these in a lot of states of decomposition, so I'm going to show it to you what's actually inside of these. Okay, my friends, I show this all the time, which is a 2-inch wide hair follicle. And that right there is the erector pili muscle. That right there is the sebaceous gland, the hair shaft, the root ball, and these two little dots are the papilla. And of course, this is the ball right down here. Now, we're going to look at this in a microscope in extreme detail, and then I'm going to give you a treat, because I haven't done this yet on this, which is a catalase test to see if there's still enzymes in here. And we're going to do it under the microscope and see if we can get that foam coming out from where the membrane's attached to the muscle. And maybe over here too. Wherever there's a membrane, wherever there's a separation, there's going to be a barrier, and that barrier is saturated with catalase. I found it in virtually everything. But this is so highly invaded, you can see it's virtually all the same material. So that's the key. Was it in an environment where everything was like basically washed out of it? Or did that catalase stay because it's locked inside of a membrane? I have found them locked inside of the membrane, still there, even though everything else was so transitioned, you'd think nothing could have survived. And I think they will be in this right here, right where those membranes attached to that muscle. All right, you're going to have to go through a little torture to get there, though, because I'm going to put it under the microscope. We're going to look at what all these little things are, and I think we can even see the blood vessel, because there's going to be blood vessels that service the muscle. You can't have a muscle without blood. It just doesn't work. So there's going to be, and I already saw them, I'm pretty sure, as you will see, uno momento. 2 inches wide. That's pretty good size. Now, I have other giants here on my property. This one hasn't been DNA tested, and I don't know what it's from. But the one I did have DNA tested was a finger. It was no question whatsoever, a fingertip. Still has a fingernail, still has the fingerprints. I drilled deep down inside, got blood, and it's excellent quality. Mitochondrial DNA is Homo Sapien. Not Denisovian, not Neanderthal, not whatever they want to call them. Modern human being, Homo sapien, mitochondrial DNA. I'm going to go through the DNA sequences and how it was tested, the PCR test, what it does, how it amplifies, how it rules out the fact that it was contaminated. What does it indicate? What is mitochondrial DNA? Where does it come from? How does it know that it was a human that did this? How much of a big chunk of that DNA do you need to prove the point that it is from whatever species that you know, and all of this breaks down to genomics. You have to know where these blocks of these genomes exist, and then you have to have something they call primers and so forth that clip off these pieces. Then you amplify them. There's a lot to it. I'm going to show you the whole thing. I know, trust me, I went through this whole thing. I don't miss much when I'm doing my research. I want to know every detail. If I don't know the details, I can't make the claims. And I make a lot of claims, but I have a lot of details, and you're going to see them right now. All right, my friends, I know as much as anybody about this. I'm going to tell you that right now. I'm not bragging, because it's true. This is a DNA extraction, which I did. I took the deep from inside. I drilled inside of them, and I got a very excellent sample, not contaminated, from arteries. Very clear. It was blood, basically, almost. Now, and so I extracted it and I sent it. He didn't extract it. The people from Helix Biolab, excellent people to work with. Fabulous job. And this is 10 years ago. It was very, very severely fought back against. And I will show you the different specimens that I had. But this is mitochondrial DNA. That is not. There's another one called a nucleic DNA, which is the male side. Mitochondrial DNA is basically from the maternal side. It's a lineage. And if you can find certain chunks in the genome, in that sequence, which is the DNA, and if you find certain chunks that have all of these different characteristics, it means it's a Homo sapien, or it means it was whatever. But the Homo sapien is what ours came out to be, mine. And it was successfully amplified. And this is PCR amplification. They take the product and they amplify it using these, I forget what they call them, but primers. All right, and they used these different primers and they amplified it, and they got excellent quality DNA from a 36-inch tip. That's a huge fingertip, which had the fingernail and the fingerprints. Excellent quality DNA. And from the lung, excellent quality DNA. And the other one was a little more transitioned so that it didn't have as good a quality. The other one was just literally raw blood. You wouldn't know the difference. Now, the 36-inch tip and the other one both, the lung, are Homo sapien mitochondrial, mother's side, cytochrome B gene, and D-loop region, Homo sapien. And I said, what does that mean? He said, it's modern human. Modern human DNA is the same thing. I said, just like us? He says, yeah, the mother's side was just like us. And he said he would stand by these results as long as you understand I am the one that extracted them. If I gave him something bad ended up. If I did a good job, it's a good job, and all of this stuff is accurate. And I did do a good job, and the negative controls were negative, and all of these sequences were correct. And this was the lab director, Tom. He's a fabulous guy to work with. He was very, very good. And you know, this is a serious test. I wanted the best. And nobody else had done one of these before. This was the first one. And absolutely the first. Well, I don't know about that. Let me just back off from that statement. What I know about was there was nobody had done DNA on ancient pre-flood creatures, which basically this is all pre-flood stuff. Had they done ancient DNA at 2015? I don't think so. That's 10 years ago. I didn't see any. I know they were looking into soft tissues and how it solidified and so forth, which is Yale did a paper after I gave them all my research. They did a paper, this one right here. Exceptional preservation of soft-bodied creatures promoted by silica-rich oceans. Exactly what I gave them in 2015. This paper goes in 2016. And they talk about it in the Yale News, how they figured this all out, and and these absolute flawless, and it was worldwide, the creatures were preserved, and they claimed this was before there was even bones or any kind of creature's bodies. It was just slimy stuff, which I know they know that's not true. I presented all the stuff, Derek Briggs, over and over and over for years, and then he's an author on this paper. I don't think that's right. So you saw, or you maybe didn't, but that's the hair follicle. We're going to actually see if there's any catalase in it. But I've shown all this stuff so many times that nobody can get their head around it. Even like Bennu. Bennu is heart muscle. That's heart muscle from asteroid Bennu. It's brought it back here on Earth. It has all the chemistry, has the sarcomeres, it has the membranes, the phosphates, it's got everything. And it's got all the chemistry, nucleic acids and DNA and the whole 9 yards. And they just can't accept what is there. And it is a heart. You can see it's a heart. These are the valves. These are the valves of the heart. And the plumbing pops off because of the iron banding when they die. Your psyche, same thing as a heart. They cannot accept the reality of the situation we find ourselves in. That's a tube coming out of that heart. That's not a crater. That's a tube coming out of that heart. That's the structure of how they sew themselves into the body, basically. That's tendon, which anchors the muscle so that it can flip and flop like that and you can pump your blood. And all of this was done from the National Science Foundation, Smithsonian. They want to really understand this. Well, I've been trying to get through to somebody there, and I met with a stone wall. And those are hearts. And here's what they look like. That's a heart. Those are the valves. Same valves. Same valves. And they break right along this line right here because of the red and the blue. There's a line of red and a line of blue, and it pops apart because of the polarities. I know this stuff pretty well. I think I deserve a piece of the table here. These are muscle sarcomeres. Those are from a heart attack victim here on Earth. And here's what it is from Mars, from Bennu. It's the same sarcomeres. And it has the same membrane layers, layer sarcomeres, layer sarcomeres, layer sarcomeres. And then you can see down into here, they form bundles. That's a cardiac muscle bundle. All right, but they all are made out of sarcomeres. So, I don't know what else I can tell you. I would like to see some reality, especially in our space programs. If they're just going out there walking around and I don't know what they're doing out there. They're just not paying attention to the details. Even Mars, this is nothing more than just gigantic biology as well. Everything is made out of gigantic biology. It's just a fact. You gotta get over it. You want to live in the truth, you got to accept the truth, or at least listen to something. It could be true, but to admit it, you break your mind. Well, you might, but truth is it's a hard thing to accept in some cases. Well, in all cases. In this case, this is not an easy truth. Like I said the other day, or maybe even today, this is the biggest mind shift in human history. Tell me something that wouldn't be more shifting of the mind than what I'm presenting here. That everything is made out of geology, and biology is nothing more than, geology is nothing more than hard biology, and hearts in space can be 140 miles wide. And that was blown right off of the top of Mars, I believe, or in Wars in the Heaven. Every culture had the same story. Gods, wars in the heavens, there was giants in the earth in those days, more giants later, battles, great flood, cataclysms. Now, if you can deny those claims, I'd like to see how. This is Mars, which was whacked by Venus, and then exactly what they said happened in the ancient days. They said Venus smacked into Mars, and it almost hit us. We escaped mostly, and then recouped, and did not learn our lesson. Mars just didn't. Mars was the god of war. You want to start wars? Well, maybe not. Maybe we should think twice about those kind of things. That's a god of war. Now, who would be considered a god of war now? Who knows? We live in a biological universe, my friend. It's time to wake thee up. All right, let's just talk about the general construction of the human genome and what genes are, basically, and what they consist of. What are they made out of? They're inside of our DNA. And our DNA, like I said, it's a program. So, what is genomics? That's a study. Genomics is a study of the entire genome of an organism. It means all of its DNA, including all of the genes, which are the code for proteins, non-coding sequences, they don't know what they do, unknown functions, structural elements like telomeres, centromeres, so forth. Unlike genetics, which often focuses on individual genes, this gene controls this gene controls that. Well, yes, they know a lot of that now. So unlike that specific genetics where it's very, very specific, it often focuses on genetics focuses on individual genes or inheritance. Genomics looks at how all genes work together, how all they're interacting together. That's what I look at, and how the whole genome affects health, traits, evolution, and disease. So we know we have a genome, which is nothing more than program. How does it get programmed? And how does it adapt to new threats and so forth, and new situations it's under? And how does it react with stress and invasion and pollutants and all of that stuff? Because this is the sequences of your body doing its job using that genome. All these different little sequences have a meaning and a purpose. Now, there are approximately 3.2 billion base pairs in the genome. Base pairs just is a little tiny snippet. And to make a gene, which is a little bit of a program, and they can be extremely complicated. It takes about, they say 27,000 base pairs in that chunk of DNA to do a pretty sophisticated job, like your arm or your leg or your eyeball or something like that. That's what genes do. They control that stuff. That's why they say, oh, he's got a genetic problem. One leg goes this way or that way. There are genetic sequences that are just wrong, and they carry out down the line. So every kid has the same problem, basically. And that's a genetic problem. That's a gene sequence that's inherited. A lot of things, the genes get damaged, or they don't turn on, or they don't turn off, or they turn on too strong, or they don't have the enzymes to work with them. All right, and the other times when you get invaded by something, you don't have the programmable bits and pieces to attack that invader. And that's also bits and pieces of a gene. They're bits and pieces of code. And they get inserted into your own DNA to make you immune the next time. From when that sequence comes in, it says, I know who that guy is. He's no good. Kill him. And that's what that does. And now it's in your DNA once it gets programmed in there because of a prior infection. Now, if you couldn't respond to that prior infection, you were so weak that you got infected and you couldn't respond, that's when you get deathly ill. And you can die because you just get overwhelmed. But if you have enough resistive bacteria and enzymes, because you're going to be programmed to be able to resist, you got a whole ton of response mechanisms that can work together. This is genomics, a genome. The whole genome works together. and you can attack. It's not just one gene. So you have to have all these little pieces in place. You got to have the vitamins, which are like coenzymes. They're not quite enzymes, but they have to work with an enzyme to do these very exotic work. This gets pretty deep, and it gets into a lot of chemistry, and you have to understand dipole electron flood theory, which works with the half-lives of all of these particles. That's why enzymes do, like they said, without an enzyme you fall over dead. I mean, you're just going to fall over dead. You don't have enzymes in your body, there you go. But if you have the enzymes, they work so fast, work in half-lives, and they go all down the line, and they do, like they said, some of them can do millions of years worth of chemical reactions, breaking and making of all of these different particles. They can do millions of years of making and breaking bonds in less than 1 second, and end up with a product that is the result of the enzyme. Without the enzyme, you don't get the product. You don't get the product, it doesn't have to, you can't get the thing done. Your muscles are weak. You can't make muscles. You can't make bone. You can't make membranes. You can't make fluids. All kinds of things happen to you when you can't absorb the correct products from what you eat, and that's in your microbiome now. They're just very, very, very deep into this now. They understand now, finally. You don't have, and Hippocrates said this thousands of years ago. He says all disease starts in the gut, and it does. Okay, so they're pointing to DNA analysis of this dragon man giant skull is actually Denisovan, which is not Homo sapien. Okay, my friend, this is a 2-inch wide hair follicle. Now, this, of course, is the hair shaft. Let me show that. Now, what are we looking at here? Let me stabilize it, because it's hard to stabilize these things. Now, this is the hair shaft coming up. All around there is like basically mud, but you can see the shaft. Now, I believe it has a flat side, and I'm told that flat-sided hairs are from curly-haired people. That's what I'm told. Now, let's follow this down the bulb, because this is a hair shaft here. All right, so there's the shaft. I'm going to come down the side right to here, and what is that? You come as far away as I can get from this. Let me hold in on it. This is really a long distance. It's 2 inches wide. That is what's called the erector pili muscle, and that is the center attachment to the muscle. It runs off this way, and it pulls the hair back and forth. Don't forget, there's the shaft, there's the erector pili muscle. That's on one side. On the opposite side, as you will see, is the sebaceous gland, which is right there. All right, you see it? That is the sebaceous gland. It's sort of drooped down the side of the root ball. Quite easy to see, though, when you stop and think about it. That's the sebaceous gland. And again, the hair shaft comes up here. So the sebaceous gland is on this side. On the opposite side is the erector pili muscle attachment. Now, I have never tried to see if there's catalase in this, but if there is, we will see it come out from right around the borderline, which are membranes. That's where the catalase is dense. Those are the fluid-filled highway, and they contain the catalase to keep extra oxygen out of your body. Those are invasive, and these are anti-invasive catalysts. Now this would be interesting. It's 2 inches wide. If the catalase is still there, that means it has survived as an enzyme over, I would say, this goes back 3,700 years. That's my estimation now. So what do we have? We have the rectopili muscle, we have the sebaceous gland, we have the hair shaft, and at the bottom we have the root ball coming right to the bottom, right there, and then it rolls right up here as to where it begins to issue from, which is right there. And these are the vein and artery. Of course, they're everything. This is very heavily transitioned. You can't really see any biological signatures other than the obvious anatomy and the rectopili muscle. Anytime you have a lot of membranes and a muscle attachment, you're going to get that. Yeah, and you're going to be able to. I'd be very surprised if there's no catalase in here, because that stuff can last, I think, just about forever. We shall see. We're going to have to try it. I'll have to come right up on top of it, because it will come out as atomic bubbles. And I'm serious. Atomic. It'll look like almost like there's no bubbles there, but they are there. It'll look like a sort of whitish looking. But the bubbles will congeal, and then all of a sudden you'll see bigger bubbles. If it has enzymes, I'll be surprised if it doesn't. I've done a lot of these, and well, not a lot of the hair follicles. But anyway, that's a good size hair follicle. A lot bigger than yours or mine, and that's a sebaceous gland right there. So I've been showing this puppy for a long time. And this is only this is the biggest thing I can hold in my hand and still prove it's an anatomical from some big creature. I'm not saying it's from a human, but it might be able to be understood, because in a muscle investment, you're going to have a vein and an artery, and that's probably where they are right there. You see this? Those two spots. When we get up real close on it, we'll take a look better. But there's going to be a vein and artery servicing that muscle. I mean, it just is. Once you understand anatomy and biology and the colors, and I mean, I'm pretty sure that's where it is. Well, we'll find out. But that's a good sized hair follicle. Okay, my friends, I made some statements that we were going to get catalase out of this, or be able to see that there was a catalase reaction in the membranes surrounding where the muscle attaches. There's a muscle that attaches to this hair follicle, and it's right down in here, and there's a couple of blood, and I make this statement, oh, I know for sure there's going to be blood. No, I couldn't get any catalase reaction to speak of at all. So I was wrong. This is so completely transitioned, there was no catalase left. That is basically an anomaly. When you get a good fresh mud fossil, wherever the membranes are, and they attach to something, if you can get down in that crack, like down in here, you should get a good catalase reaction. I got nothing. So I made a couple of statements there that didn't pan out. My tests were for the mitochondrial DNA, and I asked if mitochondrial DNA proves Homo sapien, and is it in human DNA, and they say yes, mitochondrial DNA is human DNA, and it can help determine whether a sample belongs to a Homo sapien, which is modern human, which is what we have. But it has limitations, and it's not the only evidence scientists use to classify a human species. Well, as long as the mitochondrial DNA is there in the sequence, I don't care about the rest of that stuff. What I wanted to know, and this is exactly what I told Tom at Helix Biolabs, I said I want 100% certainty that this was either a human or not a human. And if it was a human, if you can tell me what kind of a human, that would be wonderful. But is it or isn't it? Am I just crazy, or do we have DNA here? And it was dense, excellent quality. And I've shown over and over how I extracted it. And it comes right out of the actual this lung, I don't know if you can see from there, there's red blood. I could still get red blood right out of there. What I did was I drilled into the reddest spot and got the blood, literal blood. I mean, it was just blood. And that one there was good, and the big, huge fingertip, I broke off a piece and got down inside, and fingertips are saturated with blood, so that one also was dense and of excellent quality. Now, the other one was from a hand and this finger. This is a fingertip. This was CAT scanned and so forth, and DNA, but see how it's all mud looking? It's just a lot of transition. But I did drill into these things. I don't take it off the surface. And I have the whole hand for this, and the palm, and the knuckles, and bones, and all kinds of stuff. And that one there, I would say that's probably 50 feet. The big one, I go almost 200 feet. Now the 2-inch hair follicle, I don't know. I would think, I really don't know. You have to do the calculations. But I think a normal hair follicle is about 2/1000, maybe 3/1000 of an inch. So that would be a lot of times bigger than ours. A lot. All right, this is interesting to note. The limitations on mitochondrial DNA, it says you could have a Neanderthal father and a Homo sapien mother. So a mix. And your mitochondrial DNA would only show the Homo sapien line. So who knows what these gigantic creatures were. But they did have the Homo sapien mitochondrial DNA, so that's really all I really cared about. So it doesn't give the full genetic picture. Now, other human species had different mitochondrial DNA. So some of them did not have the human mitochondrial DNA, but were closely related, such as Neanderthals, Denisovians. They had distinct, different type of mitochondrial DNA, different from modern humans. All right, so don't forget, we got a modern human DNA. That's all I can tell you. And it was dense, and it was of excellent quality. And the report is online. You can look it up. All right, let's keep an eye on this. I'm going to get a little water going on there. All right, you see it? It's actually bubbling as it's going down inside the cavity. And these are one or the other. Is one's vein, one's the artery. Now, we should be able to get a good catalase reaction out of this. Not necessarily here. I've been finding the best spot is at the membrane attachments, which are going to be where the muscle attached. They're going to have little membranes around them. Are going to be excellent, and there's going to be a lot of catalase reaction in the blood. But the other ones really look cool. Let me show you. All right, take your time and think. That's the heel bone, and that's that little nubbin that's underneath everybody's heel. You got a little calcaneus spot that just is hard. That's it. That is the strap that would run up to the bone, which is the tibia. That's your main bone from your leg that comes down, and then this strap would attach to that bone and hold it together. It's a tendon tension. It's just tight. Well, when they snap, which it did, it creates a wrinkle zone. That is the wrinkle zone. That's where it's nice and flat. That's where it's all wrinkly. You see it? That's all the wrinkly stuff right above the flat place. And that's the bottom of the heel right there where it straps around and pulls that heel up into your tibia so that they stay attached and you can walk, and it's a pretty nice design. It really is. And this is the cradle that the bone sits in. And the same thing with mine has the same thing here. So that's the cradle mine has right there. And it has the bump on the bottom, just like they all do, like that one does. And it comes up around, and this is where the tibia, which is the main bone, sits right there. And the fibula was here, and that's the one that will do this and that. But in the flood, they just fall off because they only have tendons holding them on. There's just no real serious attachment. That's why you can float around so well. Anyway, I think this deserves to be looked at. I drilled into these. See that? I drilled into that to get DNA to take and send off to get tested, and they wouldn't do it. Nobody will do this on this kind of stuff. Well, I can sort of understand it. There's so much of a pushback against it that once you expose yourself, then you're a target. Really, it's unbelievable. Unbelievable. People just don't want to know the truth. So what do we see here? We see two springs. We say, well, what do you mean by that? There would be some form of toe-ish things, or whatever they are, that this would hit first. So you're standing here and you push your foot down. So let's say this is your foot, and these were inside this. You'd hit down here, and this edge up here would twist up, and that's wrinkled, see it? That would twist up this way. What would that do? There's a pin here. It would go, and this whole thing would pull this way. And there's a latch here with a strap. It comes up to this one right here. It looks like to me. That's what I'm seeing. And there's a pocket right here to give it, because when this comes here, it's going to pull this. It's going to pull this in. So your spring-loaded here, first of all, wants to come back because it's unwound. It wants to come back to its original position. So it's unwinding, and as it does, it pulls this in. So this one pulls in. Now, if you're hitting hard, you got both of these, and as soon as you let go, and it comes off the ground, it's going to straighten it right out by itself. No muscles involved, basically. There's really no muscles here. It's just basically springs. I mean, it's stunningly simple. I don't see any muscles, do you? I don't see any bones, do you? No. I see a calcaneus heel. I see a tendon. I see a place for a bone to cradle in there. Yes, I do see that. But I don't see any bones in the foot, and that's not standard. A foot is just saturated with bones. I'm not the only one that has these things. They're everywhere. That's a foot. I'm not even sure where it is, but I can tell you that is a foot. And this is, see these this here? Those are toes that are not like our toes, I don't think. And then they have these like hard things underneath. And I think that's what is for the Notoes. I think that's what the Notoes do. They have hard things underneath. They come up and they force that. Let's go back to looking at that spring load. I think they come up and they force the spring load to come up, and then it automatically comes back. Now, up here you can see this is where the tibia comes down. That's where that would have rocked right there. Now, I believe this is where the fibula attached to the side, and that I think, I'm pretty sure, is a strap. You see here and here, and it would run up to the bone, something like that. I really just don't know at this point, but it does look like a strap, and that looks like a bone, and this looks like where this red blood is coming out of there, because that's what grows green things very well, and that is a giant foot. They're not uncommon. It looks like most creatures had feet, and that's what they look like. And they were, I believe they were sealed in here, but they have eroded and opened up between it where there was some kind of toe-ish things. And when I say toe-ish, don't forget we're looking at those little hard things right there. Well, let's go back to this. If those little hard things were right underneath here, they would push up against here. It's exactly what they do. And there was a little crease around the toes, which would just let them sort of go up like that. So this would pull right around on that pin. And there's no other option other than for this to come along with it, because I believe they're attached. And you can still move this against this, but it's going to come this way. It's all pinned together off that spring. When that pulls in, this comes in, fills in this cavity. It's hard to deny what you're seeing there. And then when you just, as soon as you lift off again, your foot would just automatically straighten out. You'd be all set for the next one. No muscles involved. Very, very intricate design, basically. All right, so they're making a big splash about this Denisovian guy. I have, all my DNA tests were done 10 years ago, and they're very, very sophisticated PCR, and they weren't cheap either. And all of this was done and just refused to be discussed. Now they're starting to talk about a dragon man. Well, and then they're saying it was a giant skull and all this business. But now it's going to be in the news. Hopefully we can start to look at the ancient stuff that I have here. It's because my stuff has already been validated, 100%. DNA, CAT scans, specimen, tests, chemistry, anatomy, it's all here. And I believe it points to either new species or I'm not sure what. But I know for a fact, if you drilled in here deep enough, you'd get fresh blood out of there. And I did. I drill in. I drill down inside. And the toes, whatever, if they have toes, would have the most blood. So I don't just swab it off the surface, but I can't get anybody to test it. I had guys that wanted to pay for it. And I said, because I'm done paying for things. I paid for all the stuff. Nobody pays any attention. Why should I do that? You know, it's been 15 years I've been doing this. And it's, you know, I'm just going to say it. People want to see it, they see it. They don't, they don't. But I know it's being pushed back against, and I'm being suppressed because I'm being considered a pseudoscientist whack job, because nobody will take the time to look at the stuff that I present. It's just their mind is so closed, and that is the mindset of academia. It's absolutely stunningly closed. And they laugh at people that say the earth is flat and they can't see it. Well, PhDs, they have PhDolia. They can't see what's in front of their face. And I'm not kidding you, it's stunning. After this amount of years and this amount of pushback, but at least now, there's determined as giant skull and well, let's find out. Giant skull from China. Let's look into it, which I think I already did and showed you, but I'm going to stay on this because we need to see reality about our ancient past. Right now, it's absolutely not correct. I don't want to be nasty, but it's not correct, and it's not, nobody cares it's not correct. It seems like nobody even cares. Hard for me to believe that this can go on for this long and they get away with it, especially when they're charging the kids. If I was a kid, I'd start a class action lawsuit and say, you guys should have known about this. You should have paid attention. You should have looked. There's no denying this. And you're taking our money and saying you're acting as a good fiduciary and we can trust everything you say, and it just has not panned out to be that way. And I have all the evidence showing that. I've been showing this for way over 10 years to the people that are the ones that communicate with their kids, the Ivy League super guys. They're the gatekeepers. You don't get through them, you're done. And I can't get through them. I was done. But I just never gave up. I just kept doing it and doing it and doing it. And now, sooner or later, and I think it's the sooner right now, because people just are getting fed up with it. Fed up. Absolutely fed up. And if you're not thinking. You're just not thinking. All right? Thank you.